So I'm going to stand here. Can you hear me at the back? Is that? So, as you're well aware, I hope, last year was a very big year for people who were interested in evolution and Charles Darwin. It was the 200th anniversary of his birth in 1809, and 150 years since he published the seminal work, of course, The Origin of Species. And during last year, there was lots of you'd have seen lots of TV programs and read articles and radio shows about the life of Darwin. And you'd have heard some familiar things. So he was born in 1809. For 5 years, between 1831 and 1836, he went on this voyage around the world, the Voyage of Beagle. He was in Sydney for a few weeks in January 1836 on the way back. 1859, he publishes The Origin of Species. There were other books, very famously I'll discuss in a second. 1871, The Descent of Man. He died in 1882. He had no genetics. That was a kind of gap in his, not for his own fault, but he had no genetics. That was a gap in his work. Mendel was living at the same time as Darwin. Mendel was kind of unknown. Mendel's work was rediscovered in 1900. And in 1920s onwards, the science of genetics and selection, Darwin's work was kind of melded into what's called the Neo-Darwinian synthesis. This is a kind of familiar potted history of Darwin and Darwinism. Now, I work on viruses, you'll see that in a second, and I was interested during the kind of Darwin year to wonder whether Darwin had ever worked on viruses or thought about viruses. Darwin worked on an amazing number of things, but did he ever really discuss or consider viral diseases? And the answer is not quite. So as you'll see now, this is a history of virology that I've put up here. They're a little bit out of sync. So very arguably, very stretching a little bit, virology kind of starts, you can argue, with Edward Jenner, the English doctor who in the late 18th century invented vaccination. He didn't actually really invent it. It was well known in the Islamic world and the Chinese world, but Jenner's very famously used cowpox to vaccinate against smallpox. But he didn't know what caused it. He had no idea what the cause of smallpox was at that time. Louis Pasteur, 1885, so jumping on a century or so, very famously made a vaccine against rabies that we still use today almost. Pasteur never knew what a virus was. He never saw a virus. Okay, he didn't know how he made his vaccine, but it did work. It wasn't until 1898, some 16 years after Darwin died, that we discovered viruses. The first virus discovered was a thing called tobacco mosaic virus, it's a plant virus. So Darwin never saw viruses. He never heard about them. 1900, a very famous story in the history of virology, the vector for yellow fever, the mosquito, Aedes aegypti mosquito, was discovered in Cuba. 1918, I'll discuss this in detail, a global pandemic of influenza maybe killed 50 million people. It's debatable, but a very large number. And from 1920s onwards, at the same time as the Neo-Darwinian synthesis was going on, viruses were being discovered. So modern virology was really born in 1920. We started to find the agents of the diseases. So 1918 flu, it wasn't until the 1930s we knew it was caused by influenza virus. So Darwin never saw viruses. And I looked at Darwin's work last year to try and find had he actually mentioned viral diseases, not knowing what the cause was, but things we now know are caused by viruses. And he does. So in one part of The Origin of Man, he discusses yellow fever. It's the only viral infection reference I could find. And there's a good reason why he discussed yellow fever, because in the 19th century, yellow fever was a really, really big problem, particularly in the Americas. I live in North America now, and if you go to the south of North America, you can see lots of traces of what a bad epidemic yellow fever caused during the 19th century. There's a little martyrs kind of memorial in Memphis. And you really can't see it very well, but what it says is, in 1878, there was this epidemic outbreak of yellow fever, and 30,000 and 50,000 citizens of Memphis left the city, just ran away. Of those that stayed, 4,000 of the 6,000 Caucasian population died of yellow fever. It's a very large number. But less so, 900 of the 14,000 African American people died of the population. And Darwin saw those facts. He saw that people of African origin were not dying in such large numbers as Caucasian people. He saw in West Africa, the colonialists in West Africa were dying of yellow fever, but not the African American people. And Darwin speculates in Descent of Man that yellow fever then probably arose in Africa. It's an African virus that then spread to the Americas from the slave trade. Okay? So with the wonders of modern molecular biology and phylogenetics, you see in my talk, we can test now whether Darwin was correct. This is work done with Alan Barrett and Juliet Bryant from the University of Texas. And what we've done is sample yellow fever strains all over the world. This is actually a small tree we've done. We have bigger trees. And you can see this phylogeny and the way we've dated it fits absolutely beautifully with Darwin's ideas. This is a tree of yellow fever virus. And you can see that the oldest, the deepest split is between African viruses. East Africa here, West Africa there. So this is an African virus. The American ones, this is a small sample, we have lots more. The American strains are most closely related to those from West Africa, suggesting they came from West Africa. Of course, the slave trade was about importation of people from West Africa. Now, we can also do these days is we can date how old these trees are using things called molecular clocks. I haven't got time to explain how we do this, but the rate of evolution is sufficiently constant, you can actually go back and infer when things happened in the past. If you do that, you can see this time scale is very recent and accords very, very well with the history of the slave trade. So the origin, the kind of origin time, sorry, it should be actually this point here, the origin time of the American strains is exactly when we thought the slave trade was, 16th, 17th century. So modern molecular biology has told us exactly that Darwin was right. And I've had this long preamble on Darwin and yellow fever because this kind of exemplifies what I want to do today. I want to show you how you can use modern molecular techniques, sequencing, phylogenies, to understand where viruses come from, where epidemics come from, and hopefully how they might be spreading. Okay? And if there's one theme, I think Steve's mentioned already, there's one theme you take home from this lecture, is that there's a growing power of tree building phylogenetics and genomics to reveal amazingly detailed things about the origin and spread of viral disease. That's my kind of theme for the day. Okay, so the first part now is about how viruses emerge and spread. So when HIV first came along, discuss HIV in a second, in the 1980s, what many people did was make lists of viruses that they thought were emerging. So i.e., by emerging I mean it's newly appeared or it's newly spreading. Okay? And here's a list from 1990. I think I've used this slide since 1990, I should say, of a list from 1990 of the viruses people thought were emerging, so newly appeared or newly spreading. And some, I'm sure you'll know, HIV, Ebola, yellow fever, discussed already. Some, I'm sure you've never heard of, Maputo, Kasuna Forest. Some you probably can't even pronounce, Nyong Yong, Oropush. They cause a variety of diseases, often hemorrhagic fever. They're generally found, not always, generally found in tropical, subtropical regions. And critically, the last column here, they have a variety of animal hosts. The natural host is a different animal than the human. Okay. So that's 1990. Since 1990, we discovered a number more. This is just a list of the kind of the major ones that impact human health, but there are lots of others. So hepatitis C virus, a massive, massive killer. Sinombre, now those of you who know Spanish will know this is a very, very odd name for a virus. I can explain after if you're interested in why it's called Sinombre. Nipah hendra, that is in Australia. West Nile, of course, I mentioned a bit today, SARS. And finally, the current kind of bogeyman, H1N109 influenza swine flu. Okay, looking at this now, if you look at this list in more detail, you start to see a few trends. The first two trends are, one I've mentioned already, is that all these viruses have an animal reservoir except hepatitis C. Now, this is a real mystery. Hepatitis C infects 180 million people globally, but we don't know where it comes from. It must have an animal reservoir somewhere, but what it is a mystery. All the others have an animal reservoir, a bat or a rodent or a bird or a primate, that then jump into us. Okay. Second, all of them, except that one, monkeypox there, they're RNA viruses. So their genome is made of RNA and not DNA. And that's very important, which you'll see in a second. The second thing to see is that there's a distinction between viruses that I call spillovers, so that they only infect one person, then burn themselves out, and others that kind of keep themselves going. So, for example, rabies is a spillover infection. If you're bitten by a rabid dog, one person's infected. That person does not normally ever infect anyone else. You don't bite each other very frequently. So rabies is a spillover, well, hopefully not. Rabies is a spillover infection, okay? Most are like that. Most of this list are spillovers, no onward transmission. A few cause local outbreaks. Like Ebola, very famously, appears in villages in Central Africa, infects a few hundred people and burns itself out. It's a local outbreak. There's a few of those. The smallest number are those that have jumped species boundaries and successfully established endemic infections, become a real pathogen of the new host. So, for example, dengue is, HIV is. Yellow fever really isn't. Yellow fever is actually a monkey virus that only causes local outbreaks and burns itself out. Very few become real human pathogens. And the fact that there are three kind of groups here tells you there's an evolutionary process. It's easy to be a spillover, a bit harder to become a local outbreak, and very hard to become a new sustained infection, a new host species. So it's an evolutionary process from that to that. Okay, you have to evolve to adapt to spread of new hosts. Okay, that's very important. So now this is kind of general stuff. Now I want to explore these points here in a bit more scientific detail. I hope this is not too dense for some people. Here we go. So, first point then: viruses come from human viruses come from other animal species and jump into humans. And the kind of poster child for this is HIV. Here's a tree I kind of made for the book that I've just published. And what I've done here, so HIV has relatives in African monkeys and apes. Lots of African monkeys carry viruses called simian immune deficiency viruses, okay? And the human viruses, I put a little human symbol where they are. So all these viruses here are found in lots of African monkeys and apes, and the ones from the human symbol are from humans. So African monkeys and primates carry lots of these viruses that look a bit like HIV we have in humans. And these viruses have jumped to humans on a number of occasions. And the one to focus most on are this group here, and this is HIV-1, the major HIV strain, if you like. And that virus has jumped at least 3 times from chimpanzees, they're the reservoir of HIV-1, chimpanzees to humans. So one there, I've marked an X where they jumped, one there, one there, one there. So this virus has jumped a number of times from its chimpanzee reservoir into humans. Now, we also think we know why this has happened, so why this virus did this. And it's a very nice ecological reason. Most emergence cases have a good ecological story for why they've happened. In this case, it's because in West Africa of the 20th century, there's been a big expansion in the logging industry. So we've deforested parts of West Africa and used logging for timber and whatever. Okay? As we logged, we encroached on the natural habitat of chimpanzees and other primates. Okay? Not only did we do that, we started to use these primates we encountered as food. So there's a big trade in West Africa, what's called bushmeat. So this is a photograph for market in Cameroon. These are mammals for sale, it's market in Cameroon, and they include primates. So there's a big trade now in bushmeat because there's a secondary follow-on from the logging industry. And if you go and test these with kits, PCR tests, you find HIV-like viruses. Okay? And it doesn't take, so we're never going to know exactly how HIV jumped from chimps to humans, but it doesn't take much to imagine that you're preparing your meat, you cut yourself, you expose the blood of the meat you're preparing, and away you go. Okay? And that to me is by far the most likely reason for what happened. Okay? So HIV, and lots of viruses have the same sort of thing. They jump from another species into humans. For HIV, we also know now where and when this happened. So here's a tree that I made a number of years ago now. I'm really badly colorblind, so I apologize, the colors are kind of bad. But so this tree is a tree of just the HIV-1 part of this picture here. What I've done, I've color-coded all the branches by the strain of HIV-1. The strains are called subtypes. So in Australia and the developed world, we have what's called subtype B of HIV-1. That's the kind of green branches, okay? Other subtypes found in other countries. All I want you to see is the tree. The deepest branches are colored black, and they're from Central West Africa, the Congo region. Okay? And they fall deeper in the tree, and they fall deeper because they're probably from the place of origin. And here's a little map. What I've done in this map here is, not me I should say, but the shading here are places where chimpanzees live. So these colors are the habitats of chimpanzees. And that corresponds exactly to the place you find the oldest HIV strains. So this is a chimp virus that jumped to humans naturally, just a natural thing, in Central West Africa, around the Congo, Gabon, Cameroon area. We also know when this happened. Those molecular clocks that I use to date yellow fever origin, we can employ for HIV. If you do that, we think the age of HIV-1 in humans is maybe 90 or so years. It's obviously there's always an error bar, maybe 10, 15 years either side of that, but it's within the last century, so it's a recent origin. Now, I also told you back a few slides that most of those viruses that are emerging are RNA viruses. And I'm going to tell you why they're RNA viruses. Like HIV. HIV is an RNA virus. RNA viruses mutate remarkably rapidly. They evolve at extremely high rates. I'll give you some numbers in a second, just to show you how quickly they evolve. If this amount of evolution from here to here takes 90 years in an HIV virus, in a human, that would take 90 million years, that diversity, or mammal. So viruses, like RNA viruses, evolve about 1 million times faster than our own genes do, or other mammalian genes do. So they really are, it's like evolution on fast forward. And that really explains how they can make the mutations and adapt to new hosts. So now I'm going to explain, let's give you some numbers about these high rates in more detail. So basically in life, there's a simple division between DNA things and RNA things. There's one exception I'll briefly mention, but basically DNA in blue is slow, RNA in red is fast. So, and you can measure rates of evolution change in two ways. One's the mutation rate, that's the chemical error rate when you replicate your DNA or RNA. And the second thing is the substitution rate, that's how long it takes for a mutation to get fixed in the population through time. For DNA, be it a bacteria, a big DNA virus, or me, your mutation rate, your substitution rate is very low. So what these numbers mean, the substitution rate means is 10 to the minus 8 to the minus 9 substitutions per site per year. It means every 10 to the 8 or 10 to the 9 years, one base changes, so very slow rate. Look at the RNA wheel below here, and the mutation rates are massive. So here it's one mutation every time they copy their genome. Every time they replicate, they make one error. And the genomes are very small. The substitution rates are 6 logs higher than you see in DNA things. Now, the one exception are single-stranded DNA viruses. I won't go into why this is, but they have DNA, because they have one strand of DNA, they actually look very RNA-like for complex reasons. Now, the reason why RNA viruses are so rapidly mutating and evolving is the enzymes they use to replicate, okay, the polymerases, are very, very error-prone. There are two they can use. One called RNA-dependent RNA polymerase, one called reverse transcriptase. HIV is reverse transcriptase. They have no proofread, they have no repair, they don't correct mistakes, they just make them. So they are very, very error-prone. Again, that capacity to make errors allows them to make the mutations they need to adapt to new hosts. Okay. Now, I also told you that there's an evolutionary process, and most viruses only cause spillovers, and very few become endemic infections. So why, if they mutate so quickly, why can't they adapt to everything? Why couldn't every RNA virus jump to every species? So, and this is a really nice bit of biology. So even though they mutate very quickly, there's a cost to do that, to having that high mutation rate. And the cost is this, and it's called, so RNA viruses are not infinitely adaptable. It's called the error threshold. For all of life, there's a relationship between error rate, mutation rate, and genome size, okay? So you cannot have a mutation rate that's too high for your genome size. Otherwise, you make too many bad mutations and you collapse. Every time you copy your DNA, you make a mistake, and you make too many, and you can't keep yourself going, so you crash. RNA viruses are right on the edge, okay? So they have very high error rates, and therefore very small genome size. They can't mutate anymore. This is theory. This is I kind of made this graph up some years ago. Here's a paper, just recently, some people from Spain, after San Juan, did a very beautiful paper, and they produced exactly this curve. So the axes are reversed. These genome size, these mutation rates, but look at that, exactly the same curve. Here are RNA viruses, and these viroids are kind of RNA viruses that infect plants, very strange things. They're the fast mutating things of all. But look at that curve. That's the error threshold. Now, the key point is this then. RNA viruses mutate very quickly, but that means they must have very small genomes, just a few genes. They can't get longer because they make too many mistakes and they crash. So they have no elbow room. They can't duplicate their genes, they can't capture genes. They must work in this very small box. They're constrained. So they mutate a lot, but they're constrained. And those constraints then explain lots of their biology. So this is actually a very bad slide, I apologize, but here's a little measure that I've made of, this thing here is a measure of selection pressure, okay? So the lower these bars are, the more bad mutations that the virus is making, and the more constrained it is. So a low bar, more constraint, a high bar, less constraint, more flexibility. What I've done, I've taken some RNA viruses and I split them into those that have vectors, so mosquitoes, aphids, ticks, whatever, and those that have no vectors. They're passed on respiratory or sexually or whatever. And you can see by eye, and it's very strongly significant, the vector-borne ones have lower bars, they're more constrained than the non-vector-borne ones. And it makes a lot of sense biologically, because if you're a vector-borne virus like dengue, you need to replicate in a mosquito and a human cell. They're very different cellular environments. It's very hard to do. So the mutations trade off. And that then explains, this is why I made this point, a very important thing about the lists of emergent diseases. Although vector-borne viruses like dengue, yellow fever, West Nile, although they are very frequently called spillovers, they very rarely adapt to establish sustained endemic transmission because they're constrained, they're more constrained. So this is actually nice because it's actually a prediction. We predict then the vector-borne-like viruses are less likely to be new emerging ones because they find it harder to adapt to new species because they're more constrained. That's the first example. The second example of where these constraints dictate what viruses can do is how they actually are able to jump to new hosts. So in the simple rule, it's a very simple rule of thumb. One of the very simple rules of emergence is that the closer two animal species are, the more likely their pathogens will work in each other. So here, for example, is a very simple tree of vertebrates. Humans and monkeys are obviously very, very similar. So a monkey virus is actually quite likely to work in a human. The cells are very similar, cell receptors are very similar, it can do. As you go back along the evolutionary tree, it's less and less likely for a virus from a fish, say, to work in a human because human and fish cells are very, very different, the receptors are very different, they can't replicate successfully. Extreme example of plant viruses. If you go to the market in Sydney, buy lettuce or cabbage, it's got plant viruses. Don't kid yourself, okay? You can boil it to death, it's got plant viruses. You eat them all the day, you excrete plant viruses, you never get infected because they can't replicate in human cells. So a very simple rule are, the further you are apart the host, the less likely the virus has to jump in them. Now, what that means is then that we can work out in kind of evolutionary genetic terms how that jumping must occur. So for a virus from a human and a monkey or human ape, like say HIV in monkeys and HIV in chimpanzees, it doesn't take many mutations for a primate virus to work in humans. It's a very easy evolutionary ask to make. So in terms of, so if you're a population biologist like fitness landscapes, these are fitness landscapes. Here, the fitness valley is shallow, so things are very closely related. It only takes a few mutations to jump from, say, a chimp to a human. It's a very easy evolutionary ask, only 2 or 3 mutations. In fact, for HIV, they think maybe only 1 mutation is required for a chimpanzee virus to adapt to replicate humans, because we're very similar in our cells, chimps and humans. The further you are apart, the harder it is for that virus, for the hosts are apart, the harder it is that virus to work in humans. For example, so the fitness landscape's sharper, it's steeper. So, for example, avian flu, H5N1 avian flu, the current estimate is that it requires 13 mutations to make that avian flu into a human influenza virus, a virus that will spread in humans. 13. And 13 is a lot to make. So, summarize so far. RNA viruses have amazing mutational power, and that underpins their ability to jump and emerge in new hosts. The problem is the kind of trade-off that's good for us is that high mutation rate also limits their genome sizes, so they can't move very far, they're kind of constrained. Okay? And in some cases then, because of those constraints, it's hard for them to jump to new hosts, like humans, like our species, particularly if the host it comes from is a very long way away in evolution, like a bird or a fish or a reptile. It's a hard thing. Okay? So that's good news.